All right, welcome in everyone. If you're a highly motivated student gearing up for fiercely competitive medical exams like NEET, or you're prepping for your board exams, you are in exactly the right place. Consider this explainer your definitive, zero-compromise study guide for Section 5.1 of your biology syllabus, the molecular basis of inheritance. We are going to break down every single point, literally line by line, so you don't just memorize the material, but actually understand it inside and out. We want you recalling this instantly when it matters most. So let's go secure those marks.
Okay, let's dive into this. Here is our roadmap for mastering this section. We'll start with an introduction to DNA, map out the polynucleotide chain structure, explore the famous double helix model, break down the central dogma, and finally, look at how DNA is packaged in both prokaryotes and eukaryotes. I highly recommend using this exact structure to map out your own notes. Section 1. Introduction to the DNA.
So at its absolute core, DNA, or deoxyribonucleic acid, is a long polymer of deoxyribonucleotides. Think of it like a massive chain made up of millions of tiny repeating links. The total length of a DNA molecule is usually defined by the number of nucleotides or base pairs that it contains. And this length, it's actually a unique, defining characteristic of an organism. Now let's slowly walk through some highly testable statistics that you absolutely must commit to memory. Grab your pen. First, the bacteriophage known as Phi-X174 has exactly 5,386 nucleotides. Another virus, bacteriophage lambda, has 48,502 base pairs. Moving up the complexity ladder, the bacteria E. coli has 4.6 million base pairs. And finally, the haploid content of human DNA, meaning just one single set of our chromosomes, contains a staggering 3.3 billion base pairs.
Moving right along to Section 2, the polynucleotide chain structure.
You might be wondering, how does biology build a molecule with billions of units? Well, we start by zooming into the micro level with a single building block, the nucleotide. A nucleotide has exactly three components: a nitrogenous base, a pentose sugar, and a phosphate group. Let's break down this assembly step by step, like a foolproof chemical recipe. Step 1. A nitrogenous base links to the 1' carbon of the sugar. This happens via an N-glycosidic linkage, forming what we call a nucleoside. Step 2. A phosphate group attaches to the 5' carbon of that nucleoside via a phosphoester linkage. Boom! Now you have a complete nucleotide. Step 3. To link two nucleotides together, the 3' carbon of one sugar links to the 5' carbon of the next through a 3 to 5 phosphodiester linkage. This creates a dinucleotide, and you just repeat that exact process over and over to build your massive polymer chain.
Because of how these chemical units link up, they form a very distinct backbone made entirely of repeating sugars and phosphates, while those nitrogenous bases project inward. Now pay super close attention to the geometry of the chain here, specifically the ends, because they define the polymer's polarity. At one end, the sugar has a free, unlinked phosphate group sitting at its 5' carbon. We call this the 5' end. At the total opposite end of the chain, the sugar has a free hydroxyl group at its 3' carbon. Naturally, that's known as the 3' end.
Before we go further, we need to clearly contrast DNA with its close cousin RNA because exam setters love this comparison. There are two major chemical differences you need to know. First, the sugar. RNA contains ribose, which has an extra hydroxyl group at the 2' position. DNA contains deoxyribose, which literally means it's missing that oxygen. Second, the bases. While both share adenine, guanine, and cytosine, DNA exclusively uses thymine, whereas RNA uses uracil. Now, pause for a second. I want to highlight a massive exam trap. Thymine is chemically known as 5-methyluracil. If you see 5-methyluracil on a test, don't panic. It's literally just thymine.
Section 3. The double helix model.
The historical timeline of this discovery is actually pretty wild. Back in 1869, a guy named Friedrich Mischer first identified DNA as an acidic substance inside the nucleus, and he called it nuclein. But here's the catch. Because such a massively long polymer was virtually impossible to extract intact with 19th century technology, the structure remained a total mystery for decades. Fast forward to 1950, and Urban Chargaff observes a strict mathematical rule. The ratios of adenine to thymine and guanine to cytosine are always constant and equal to 1. Finally, in 1953, James Watson and Francis Crick unlocked the whole thing. Relying heavily on some brilliant X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, they proposed the now famous double helix model.
Now, what's really interesting about this breakthrough is the concept of complementary base pairing. Because adenine only ever pairs with thymine and guanine only pairs with cytosine, if you know the sequence of one strand, you instantly know the other. It's like a mirror image. This perfectly implies a semi-conservative replication model.
Basically, one parental strand acts as a precise template to synthesize a brand new identical daughter strand.
Let's methodically list out the salient features of this double helix. First, it's made of 2 polynucleotide chains with that sugar-phosphate backbone on the outside. Second, the chains have anti-parallel polarity. Think of it like a two-way street. One strand runs 5' to 3', and the other strand runs completely backward, 3' to 5'. Third, the bases pair up via hydrogen bonds. Adenine forms exactly 2 hydrogen bonds with thymine, guanine forms exactly 3 with cytosine. This architectural rule, where a double-ringed purine always pairs with a single-ringed pyrimidine, is absolutely crucial because it guarantees the distance between the 2 chains remains perfectly uniform all the way down.
Continuing with these features, the distance between consecutive base pairs in the helix is approximately 0.34 nanometers. I'm going to say that one more time so you can lock it in. The distance is 0.34 nanometers. Definitely memorize that. Finally, the plane of one base pair stacks directly over the other in the double helix, almost like steps on a spiral staircase. This physical base stacking, combined with those millions of hydrogen bonds we just talked about, is exactly what gives the helical structure its immense stability.
Section 4. The central dogma.
So with the physical structure completely solved, the scientific community needed to understand how it actually functioned. Enter Francis Crick, who proposed the central dogma. He explicitly mapped out the directional flow of genetic information in biology. Here's how it works. Step 1. DNA stores the master information and copies itself through replication. Step 2. That information flows from DNA to RNA through a process called transcription. Step 3. The information now in the RNA is used to synthesize a functional protein via translation. This directional flow, DNA to RNA to protein, is the absolute rock-solid foundation of all molecular biology you'll cover in this exam.
But right before we move on to how all this massive amount of DNA physically fits into a cell, here is an active checkpoint for you. Your textbook explicitly asks this question. In some viruses, the flow of information is actually in the reverse direction, moving from RNA back to DNA. So can you suggest a simple name for this process? Well, think about the forward process, transcription. Therefore, the reverse is logically called reverse transcription. Keep that in your back pocket.
Section 5. Packaging in prokaryotes.
So mammalian DNA spans billions of base pairs, right? If you stretched it out, it would be over 2 meters long. So how on earth does a microscopic cell package such a tremendously long polymer? Let's start by looking at simpler organisms, like prokaryotes, for example, E. coli. Even though they lack a defined nucleus, their DNA is absolutely not just scattered randomly around the cell. Because the phosphate groups make DNA negatively charged, it acts like a magnet for specialized positively charged proteins that hold it tightly together. This compacted structure is localized in a very specific region called the nucleoid, where the DNA is neatly organized into large, tight loops to manage its incredible size.
And that brings us to Section 6, eukaryotic DNA packaging.
In eukaryotes, like us, with our massive 2-meter-long genomes, this organization is much, much more complex. It requires a highly specialized structural hierarchy. At the very base of this hierarchy are histones. Histones are a set of basic, positively charged proteins. So, the crucial point here is, where exactly does this positive charge come from? Well, it comes directly from an abundance of basic amino acid residues with charged side chains, specifically lysine and arginine. Take a second to jot those 2 amino acids down, lysine and arginine. These histones then organize themselves into a neat little 8-molecule unit called a histone octamer.
Which brings us to the fundamental repeating unit of eukaryotic packaging, the nucleosome. A nucleosome is formed when that negatively charged DNA strand acts like a thread and wraps tightly around the positively charged histone octamer, which acts like a spool. A typical nucleosome contains exactly 200 base pairs of the DNA double helix. Make sure you pause and get that 200 base pairs metrics safely in your notes.
And this brilliantly illustrates the whole concept. When viewed under a powerful electron microscope, you can literally see these repeating nucleosomes. They physically appear as tiny little beads connected by the DNA string. Inside the nucleus, this repeating beads-on-string structure forms what we call chromatin, which are essentially thread-like colored bodies within the nucleus. All right, here's a fantastic mathematical challenge straight from your textbook to really help you grasp the immense microscopic scale of DNA packaging. Theoretically, how many of these beads, these nucleosomes, do you imagine are packed into a single mammalian cell? Well, let's do the math.
If a diploid human cell has 6.6 billion base pairs and each bead wraps up 200 base pairs, you just divide the two. 6.6 billion divided by 200 gives us roughly 33 million nucleosomes. 33 million of them in one single microscopic cell. Absolutely mind-blowing.
Now, as the cell prepares to divide, this chromatin coils up even further into chromosomes. This higher-level packaging requires an additional set of proteins collectively referred to as non-histone chromosomal or NHC proteins. Finally, before we wrap up, you must strictly memorize the contrast between the two states of chromatin. Euchromatin is loosely packed and stains light under a microscope. Because it's loose, the cellular machinery can easily access it, meaning it is transcriptionally active. Heterochromatin, on the flip side, is densely packed and stains dark. Because it's locked away so tightly, it is transcriptionally inactive.
Well, we did it. We've successfully mapped the entire physical structure of DNA, point by point, from a single tiny nucleotide all the way up to a highly condensed chromosome. You now possess the definitive foundation of Section 5.1. But this leaves us with a pretty massive provocative question, doesn't it? Now that we know its static structure perfectly, how does this master blueprint actually unzip, copy itself, and orchestrate the insanely dynamic processes of life? Review your notes, solidify your understanding of these core facts, and I'll see you in the next explainer where we tackle replication and transcription.